We are members of the University of Calgary's Biomedical Engineering Research and Innovation team, and we've developed a solution for rapid real-time detection of airborne pathogens like COVID-19. Imagine you're getting ready to leave for a vacation, and you're at the airport checking into your flight. A lot of people are sneezing and coughing around you, but you don't think much of it since they looked fine. No one looks sick. You carry on with your day, get on your flight, and arrive at your vacation destination. But suddenly, you fall very sick and find out days after that you contracted COVID-19. Now, your vacation is ruined. But what if you had a real-time warning at the time you were exposed to the pathogen? Like some sort of superpower that lets you know that your health is in danger. You would then have the opportunity to minimize your exposure and even possibly prevent infection, so you can relax when you arrive at your vacation destination. During the COVID pandemic, little was known about the virus, and method of detection was a challenge. It was known that individuals showing symptoms can transmit the virus through airborne particles, but it was also found that human hosts could be asymptomatic and still spread the infection. Now, we have PCR test kits for rapid detection, but what if there was a real-time detection method in larger spaces, such as hospitals, clinics, and public spaces? What if there was an adaptable biosensor that could detect airborne pathogens like COVID-19?
COVID-19 won't be the last airborne threat capable of starting pandemics, which leads to why a new technology like this air particle detection sensor is necessary.
We propose AeroSense, an air monitoring system designed to detect airborne viruses in real time. This technology offers a scalable, cost-effective, and non-invasive solution for mass screening in high-risk environments such as hospitals, airports, and other public spaces. Through AeroSense, we can identify asymptomatic carriers, allowing early intervention, and can prevent future outbreaks and alleviate strain from our healthcare system. In addition to providing large-scale coverage, AeroSense reduces the need for individual rapid tests. A study conducted up until August of 2020 found that PCR testing for COVID-19 worldwide resulted in 15,439.53 tons of plastic waste. Of this, 97% were incinerated, releasing toxic chemicals into the environment. AeroSense eliminates these environmental drawbacks by offering a sustainable method of detecting airborne pathogens, allowing individuals to take preventative measures like isolation or mask wearing without the need for invasive tests.
Additionally, rapid antigen tests are not universally accessible or free. If implemented in high-risk public areas, AeroSense could provide individuals, especially those without access to testing kits, with timely alerts about potential exposure, enabling them to take appropriate action.
By increasing accessibility, this sensor supports 2 UN Sustainable Development Goals: good health and well-being, and reduced inequalities. AeroSense has the potential to significantly reduce the spread of diseases like COVID-19, relieving pressure on healthcare systems and contributing to a more sustainable future. But how does it all work? And what is the science behind it all? We should first discuss how airborne pathogen diseases behave. Take COVID-19, for example. It is a respiratory viral infection that can cause fever, sore throat, fatigue, and in more severe cases, death. At the molecular level, COVID-19 is a single-stranded positive sense RNA virus, which allows immediate replication of virons. On the outer surface, spike proteins are in charge of binding to the host ACE2 receptors at a high affinity. In humans, this receptor is mostly in the alveolar epithelial cells in the lungs, but it is present throughout the body in many of our organs. Then, through endosomal fusion pathway or surface fusion pathway, the virus gets internalized into the host cell and the infection spreads. Understanding the viron's molecular components is crucial to the biosensor's design. Viruses and bacteria use surface proteins to bind selectively to specific host receptors. For our biosensor, we must first design a selective receptor, an aptamer, that binds specifically to the COVID-19 S protein using CellX. This aptamer is engineered to contain a fluorophore and a quencher. When in close proximity, the quencher suppresses the fluorescent signal emitted by the fluorophore. The biosensor's detection surface consists of a graphene oxide surface that immobilizes the aptamers, maximizing the binding area for detection. Let's say the air is contaminated with virons. As the contaminated air flows through, the viron's S protein will bind to the aptamer, causing a conformational change that separates the fluorophore and the quencher. The separation emits a fluorescent signal, indicating COVID-19 detection. After detection, the system can be reset through low-concentration chaotropic salts, or urea, to disrupt viron binding, followed by a buffer wash to restore fluorophore quencher system and pH for reuse. It is clear that AeroSense will require significant planning and considerations when moving forward. particularly when analyzing initial investments, packing and distribution, materials and manufacturing, regulatory hurdles such as FDA approvals and Health Canada certifications, and prototype testing costs. So the proper funds will need to be allocated towards research and development, creating functional prototypes, and validation of sensor accuracy.
What's next for AeroSense? Our design could offer solutions for other pathogens aside from COVID. Integrating a multi-aptamer surface that can detect other viral proteins and bacterial proteins could expand the biosensor's detection capability to other airborne viruses and bacteria. Additionally, the physical presentation of AeroSense should be considered. It could be some sort of tower placed in an open space, so we can move away from a handheld testing system like current breathalyzer type sensors that exists. Furthermore, certain sensor components can be changed, like our detection system using ions instead of fluorometry. This approach may be cheaper in the long run and more feasible overall. Though still in early stages of ideation and creation, we believe AeroSense can change the way pathogens like COVID-19 are contracted and spread. The sensor can make an impact in a sustainable, accessible, and effective manner.